Chapter 4, Carbon and the Molecular Diversity of Life. What I want you to learn from this chapter is definition of organic compounds and how you differentiate between organic molecules and others, and Stanley Miller's experiment, which talks about how life might have originated. Second section is about isomers, and in isomers I want you to know the definition, different types of isomers, how you can identify each one of them. Third section is about functional groups. Functional groups, there are 7 of them, and I want you to know their names along with their structural formulae and understand where they are used in a daily life, so that you can relate to what you have studied in class and understand the molecules and substances differently from what you have been perceiving them as till now. So first one, let me see if you are ready for this chapter and what we learned from the previous chapters. How is it going to help us learn more? So we are building. In biology, we always build the concepts. So most of the concepts that we learned from atoms, molecules, atomic structure, and about organic molecules such as DNA that we learned, all that will be tied together and we are going to learn more. So organic molecules that definitely contain which element? Carbon, phosphorus, oxygen, hydrogen, or nitrogen. As per definition, they definitely contain carbon. And look at these examples. Carbon dioxide, it contains carbon. So is it organic? I'll let you guys think, and I'll address this question in the later slides. And methane, CH4 is methane. Is this organic? What is the difference between these two molecules, right? So answer is A. They definitely contain carbon. And just because something contains carbon, does it become organic? That's the next question. So let's see. What kind of bonds does carbon form? Naturally, it forms covalent bonds, and that too 4 covalent bonds. Not less than 4, not more than 4 covalent bonds. And when carbon is forced to react with aluminum, then it will form aluminium carbide by ionic bonds. That is something which is not there in the textbook. I'm adding on top of what is given. And other things, yes, you can pretty much reject. Answer is E for this. Okay, you must be wondering that, when we are in a biology class, why are we learning all the chemistry and about carbon and all the different molecules, what bonds and all that, right? The water we drink, without which we cannot survive more than a day, is a chemical, and we are made of chemicals. Bacteria are made of chemicals. If you want to fight infections, we have to understand how our body works and how they work, and we have to take advantage of some things which are not there in them and which are in us. And for that, we need knowledge of chemistry. And we want to be better coming years and generations, and for that we need to know how we can genetically modify ourselves and make ourselves like Superman. Just kidding. Anyways, genetics is in progress, and it might even happen. I was kidding, but it might happen one day, or maybe you guys will be part of that research, and you will invent some new genes which were not in use all these days, and how to activate those genes we already have, or make new genes which give us superpowers. Well, benefits are plenty, And yes, we need to learn chemistry to understand all this and to make our dreams come true. And I want you to come up with an idea of making your own future. Working for an organization will be great, but just think about, having your own company which has a cool idea which you are able to manifest and make it real, and you're unique in this entire world, right? And you're making profits, right? And that will be something. So I want you to be innovative and creative and bold. So think about it. And for all that, we need knowledge. Knowledge is power. Moving on. Why carbon? Carbon is the element which is found in all organic molecules, and we are made of organic molecules. So, in one statement, living organisms, we are made of molecules which are based based on carbon, and carbon can form large molecules such as proteins, DNA, and polysaccharides such as carbohydrates and lipids, oils, many things, many molecules which make life possible. So, study of organic molecules is organic chemistry, and organic chemistry is all about carbon molecules, carbon compounds. And we are going to a little bit of history now. Um, before 1953, there were two schools of thought. One is called vitalism, the other one is called mechanism. Why are we talking about this? It's because we want to know who discovered that organic molecules can be made in labs. Who did that first? Several of them started around 1800s, but then finally who showed successfully was Stanley Miller, through his experiment, which we are going to talk about in a minute. But before we go there, let's understand what is vitalism and mechanism. Vitalism is a school of thought where scientists in olden days thought that there should be some kind of life force which makes synthesis of organic molecules possible, and organic molecules cannot be made in a laboratory whatsoever. Without a life force, it is not possible in a laboratory. Whereas mechanism is totally opposite school of thought, where they believe that no, if we try and experiment and try in a better way, then we should be able to make it possible. We can make biological molecules in abiotic environments. That means without any living organism in a test tube. So that is vitalism and mechanism. You might want to pause the video here and make some notes of proper statements of what is vitalism and mechanism. There's no point in reading those when they're already there. So let's talk about how life might have originated. Who knows? We were not there, right? Stanley Miller, he made an apparatus like this using some bulbs and tubes, and he did some welding work in order to put all these bulbs and tubes together. And he took seawater in this bulb, applied some heat, and in this another bulb he collected atmospheric gases such as methane, ammonia, hydrogen, and he started heating up water. Water boils, it evaporates. When it goes through here, it has reached the cooler area of the apparatus. In this bulb, he made sure that he put in the two electrodes and passed electric current, and they made a spark. And because of this energy, these molecules, they break their old bonds and they bump into each other, intermolecular interactions. Again, it is called intermolecular interactions. And because of that, the new molecules are formed. those new molecules which are formed in this bulb, they get washed down because water vapor which is formed here is cooling down and forming water and passing through this tube, and because this is opening here, it is getting collected here. So Stanley Miller, when he performed this experiment in 1953, he saw that this water contained organic molecules such as amino acids, formaldehyde, hydrogen cyanide, and all this. And to his surprise, he was successful, and so the world first time found that experimentally proven that yes, organic molecules can be definitely made in completely, totally abiotic environment. Previously, some scientists tried, but they took the original ingredients from animal-based products, so they were not given that credit. Well, coming back, what does this indicate? Look at this, guys. This indicates the atmosphere, right? This is seawater, the real seawater. So imagine millions of years ago when Earth started cooling down, and there were absolutely no animals and plants. There was only ocean and atmosphere, and sun's heat was causing this water to evaporate from seas, and there was lightning, and that caused gases in air to break and reform new bonds, and they made some organic molecules, and those organic molecules are maybe DNA, RNA, and some lipids, and all of them got washed down. So lipids, being hydrophobic, they formed little bubbles, and those bubbles, when they were forming, they packed some nucleotide chains, which is DNA or RNA, and that is how the first cell might have formed. So again, this is not a theory, this is not fact. We do not know, we did not exist when life originated. But through this setup, this closed system, Stanley Miller tried to show how life might have originated when Earth started cooling down and the first cell might have formed. So formation of bonds with carbon, as I said earlier, carbon can form 4 covalent bonds because it has only 4 valence electrons, and it is looking for 4 more, nothing less, nothing more. And carbon can form single, double, and triple covalent bonds. I haven't seen any quadruple bonds. When molecules are formed by carbons, we have to study them, and when we study them, we have to write those molecules on paper. So there are different ways to represent molecules on paper, and they are name, molecular formula, structural formula, ball and stick model, space filling model. Name is simply a word. A molecular formula tells us how many elements are there and how many of each element. We represent it with a number and we put it as subscript. Whereas in structural formula, we do not use any numbers, we simply draw them out. How many ever they are, we draw them out. Ball and stick model, yes, we draw them out too, but here we use circles and lines, different kinds of lines, a thick line, a straight line, a dash line. Dash line indicates that the bond is in the back. So this ball and stick model is giving us on a planar screen a 3D model. It is trying to show which one is front, which one is back, which one is on the side, which one is on top, which one is in the middle, which one is in the front, downwards. So it is giving us a 3D picture. So this is the best thing that we can possibly draw on a paper and understand the structure of the molecule. Guys, remember, structure is everything. And structural formula is not the best representation, but easy to draw compared to this one. Space filling model is not used very much, but it will give us the overall shape of the molecule. And from this slide, you don't have to memorize anything, just know what are the different ways to represent molecules on paper. So 5 ways: name, molecular formula, structural formula, ball and stick model, space filling model. And what is each one? What is molecular formula? What is structural formula? So how are they different from each other? You don't have to memorize the examples. Next concept before we go to the section where we are trying to understand what are organic molecules is the partners of carbon in organic molecules. The most frequent partners of carbon are hydrogen, oxygen, nitrogen. Organic molecules definitely contain carbon, and on top of that, they also should contain hydrogen. In addition to hydrogen, they contain oxygen and nitrogen. Remember, hydrogen has only one valence electron, so it needs one more because it's the first orbit. Oxygen has valence electron as 2. Valence shell as 2, and valence electrons are 6, so it needs 2 more electrons to make it happy because second shell requires total of 8 electrons. Nitrogen has second energy level or second shell as its outermost shell, and it requires 3 more because it has only 5. And we talked about carbon already. Carbon has second shell, which is energy level, just like oxygen and nitrogen, and it should contain 8 to fill it to the max, but it has only 4, so it needs 4 more electrons. So carbon can associate with 4 hydrogens, and they all can stay together happy, which makes it methane. And carbon with hydrogen, oxygen, and nitrogen in combination, they all can form many molecules. We'll look at them coming to the slide where we're trying to understand, just because there is a carbon in a molecule, can we call it organic? Some textbooks say that carbon dioxide, which has carbon and 2 oxygens, is not organic. Well, why? I'll let you guys look into it, but the best example that I like to give from my side is diamond. Diamond contains completely of it's a molecule made of completely of carbons, and being made of carbons, do you think it is an organic molecule? And another thing, graphite, which is your pencil lead. You call it pencil lead, but actually the chemical name is graphite. That one also is made of pure carbons, all carbons bonded to each other. But look at this, the way they are bonded to each other, one is a diamond and the other one is graphite. Are they both organic? Think about it. If you do some research, you'll find out that they are not, okay, though they contain carbon. So just because something contains carbon does not qualify to be an organic molecule. Then what is an organic molecule? Chemists have concluded that organic molecules should contain carbon, and in addition to carbon, they should also contain hydrogen. Having oxygen is not enough. They should contain hydrogen also, and on top of those 2 elements, they can contain anything more. So this is urea. Urea definitely contains carbon and hydrogen, and on top of it, it contains nitrogens and oxygen. So yes, it is organic. Is carbon dioxide organic? Well, as I said, some textbooks consider it as not organic. though we breathe it out and we make it in our bodies. The next section is about chains of carbon atoms. What is chains of carbon atoms called? When a carbon chain is made, there are several carbon atoms linked to each other. This becomes a backbone of an organic molecule, and that is called carbon skeleton. So carbon chain or carbon skeleton, both are one and the same. A chain of carbon atoms which are bonded to each other by covalent bonds, non-polar covalent bonds. And there are three types: straight, branched, closed ring. Look at the screen and try to guess how many are straight. Though this butane is under branching heading, it is not a branched molecule. So there are a total of 5 molecules on this screen which are straight carbon chain molecules: ethane, propane, butenes, and butane. Yes, 2-methylpropane is isobutane. Now tell me if this is straight or branched. If you're thinking that this is branched, it's not true. Just because they're angled like an L and they're not in straight line, do not consider this as a straight molecule. No, this is not a straight carbon chain. This is a straight carbon chain. This is not a branched carbon chain. For this to be branched, remember guys, it should have a carbon here. When a carbon is surrounded by 3 more carbons, not 2, when a carbon is surrounded by 3 more carbons, then only it is considered as a branched carbon skeleton. So T is branched, L is not branched. L is still a straight carbon chain. Okay. So these are rings. Rings can have double bonds. Straight also can have double bonds. Notice that. Hydrocarbons. What are hydrocarbons? As the name itself is saying, hydrocarbons are those organic molecules which consist of carbon and hydrogen. Where are they found? They are found in fuels, fat, which we store for later, using it later. And one thing that I want you to remember about hydrocarbons is that they are made of chains of carbons, and these carbons are attached to hydrogens, and these hydrocarbons can be part of any other molecule as well. But these chains of carbons which are also attached to hydrogens, they have non-polar covalent bonds. So look at this. This is a triglyceride molecule. So this part is glycerol. The core part is glycerol, and to this glycerol, these yellow highlighted ones, 3 chains, they are hydrocarbon chains. The black part is chain of carbons, and these carbons are satisfied by hydrogens, which are given in gray circles here. So these bonds between the carbons and the bonds between the carbon and the hydrogen, they are all non-polar covalent bonds. What is non-polar covalent bond? Non-polar covalent bond is when the sharing of pairs of electrons are equal. When they are not unequally shared, that means they are equally sharing it, then it is called non-polar covalent bond. Remember, carbon and hydrogen, none of this is electronegative, so they share equally, and that is why it is non-polar covalent bond. Whenever it is non-polar covalent bond, that means they do not have charges. When something does not have charge, then they are non-polar substances. Non-polar substances means they have no charge, so they do not like water. That means they're hydrophobic. Guys, it is all interdependent. So whatever you learn from the previous chapters, it is all going to be used, and we are building the higher concept. Remember, triglycerides are found in fats and fuels, and these bonds, these non-polar covalent bonds between carbons and carbon and hydrogen, they have a lot of energy, and when they're broken, they release high energy, and that is why these molecules are used as fuels in our machinery and as well as in cars. This is adipose cell. This adipose cell is nothing but cell which stores lots of fat molecules. Whenever we eat extra carbs and fats, they are carbs are converted into carbs means carbohydrates are converted to fats, and they're stored in inside the cells called adipose cells, and these adipose cells are found more under our skin. And these adipose cells, they contain lots of fat. If we eat more fatty food, we end up having a lot of this. We need fat, but not that much fat. We are going to talk about this in the next chapter. So coming to the next section, isomers. I bet you remember this iso prefix. Iso, previously we used this iso for isotopes. Guys, remember, isotopes are different atomic forms of same element. Isotopes has T in it, atoms has T in it. So isotopes are atoms, whereas isomers are molecules. Isomers, word has M, a letter in it. Molecules have the letter M in them. Molecules, M for molecules, right? So isomers have M in them. What are isomers? Isomers are the molecules which have same molecular formula but different physical structure. Hence they have totally different properties, because in molecules everything is dependent on its structure. First of all, what is molecular formula? H2O. When you write H2O, that is molecular formula, right? Like that, glucose can be written in molecular formula C6H12O6. Sucrose can have a molecular formula too, right? When you write. So glucose and fructose, they are isomers of each other. Like that, there are several, and isomers are the molecules which have the same number of elements in them, but the way they are arranged is different. That is why they smell different, they taste different, they react with other substances differently. So they are totally different. We think that, when you write down the molecular formula on the paper, it might seem, but we write same molecular formula, but we say that one of it would be one kind and other would react this way. So those are isomers. Remember, basically isomers are molecules which have same molecular formula, but their structure is different, hence they have different physical and chemical properties. This definition applies to all the different isomers, structural, cis-trans, and enantiomers. How you pronounce this word? Enantiomers. Just split the word. When you are not able to pronounce the word, split the word into parts which you can say, pronounce, and just put them all together. So I say this way: enantiomers. Enantiomers. Okay, so 3 different types of isomers. So what are structural isomers and how are they different from cis-trans and enantiomers? Let's lay out the basics first, and we will talk more about it so that we can understand them better. Structural isomers, they have different carbon skeleton, to keep it simple. Cis-trans isomers, they have different orientation of the groups of atoms or the elements attached to the carbon skeleton. Here, remember, structural isomers, they have different carbon skeleton, whereas cis-trans isomers, they have same carbon skeleton, but the molecules which are attached to the carbon skeleton, they are oriented differently because of the flexibility or inflexibility of the carbon chain. Enantiomers, they are the molecules which have a totally different three-dimensional pattern. So for this, to understand this, you have to watch the video that I'm going to refer to. Okay, so these are different isomers. The definitions you can get from the PowerPoint slide, guys. Moving on. So you can see these two molecules here at the top. This one is C5H12. This one is also C5H12. So when you write the molecular formula, you write C5H12, C5H12. Pentane, pentane. But this is not pentane. This is 2-butane, 2-methylbutane. So names are different, but the molecular formula is the same. And look at the structure. If one is straight, the other one is branched. And because of this, they smell different, they taste different, they react different. So those are structural isomers. You can find a lot of those in real life. In chemistry, you will learn a lot more of this. Coming to cis-trans isomers, they have carbon skeleton which has double bond, and around this carbon skeleton the molecules or elements that are attached to them, if they are on the same on the same side of the carbon skeleton, then they are called cis isomers. And look at this, if the elements are on the opposite sides, then they are called trans isomers. So this is cis isomer and this is trans isomer. Cis means same, trans means different. Look at this, they have same molecular formula, right? If X is bromine, then it would be C2Br2H, and same thing here. I will try and draw an outline of this molecule. Okay, this one looks like this. Nice poodle. Look at this. This one looks different because hydrogen is smaller and bromine is bigger. I have to do this, right? It's like, you know, this dog is running faster and this one is standing still, right? So you can see the difference in the structure. They're not same molecules. Enantiomers. Enantiomers are those two organic molecules which have a carbon. It has 4 different atoms or groups of atoms bonded to them. These two are enantiomers of each other. One is called L isomer, the other one is called D isomer. When you look at these molecules on the screen or in textbook, you might think that, you know, how are they different from each other? All I have to do is D isomer, I just flip it and turn it to the left, and it will become L isomer, right? Well, before we watch the video, I want you to learn that enantiomers are those two isomers which are definitely different from each other. They have asymmetric carbon. What is asymmetric carbon? A carbon which has 4 different atoms or 4 different atoms or groups of molecules attached to them. And having this achiral carbon, the orientation is specific with carbon. So how they are arranged with respect to those carbons, asymmetric carbons, they become different molecules. So that arrangement makes these two molecules mirror images of each other. What are mirror images? If you face your palm side facing each other, then they are called mirror images. Whereas you place one palm in front of you and take the other palm and just place it on top of it, then you can see that your thumbs are pointing away from each other and they're not matching. So enantiomers definitely have asymmetric carbon. They are mirror images of each other, and they are not superimposable. Remember guys, whether it is structural isomers, cis trans isomers, or enantiomers, they all have same molecular formula. These pairs, they have same molecular formula. Though they have same molecular formula, because they have different structures, they happen to have different physical properties and different chemical properties. And yes, it makes a huge difference. One we can consume, the other one we cannot tolerate. One can be used as fuel, while other one cannot be used as fuel. One is good for health and the other one is not good for health. Oh, a little bit more about enantiomers. I haven't finished explaining enantiomers yet. So what is asymmetric carbon? A carbon having 4 covalent bonds around it and is attached to 4 different things, then only it will be considered as asymmetric. Look at this carbon. Do you think this one is asymmetric? Yes. How about this one? Bromine, hydrogen, methyl, hydrogen again. So no, this one is not asymmetric carbon. So this carbon is not asymmetric. How about this one? It does not have 4. It has only 2 elements and 1 group of molecules. No, so it is not. This is not asymmetric carbon. How about this one? If you're thinking that CH2 and CH2 are same, so it is not. No, what I'm asking you to look at is this entire group. This is C2H5, whereas this one is C3H7, right? And this is CH3 and H. So yes, this is asymmetric carbon. This carbon, which is attached to 4 different groups, and one of which is element, hydrogen, is asymmetric carbon. So this one is asymmetric carbon, this one is asymmetric carbon. This is just information to understand which one is asymmetric and which one is not asymmetric. I want you to watch this video and come back. This video, pause the PowerPoint video and please type this up and watch this video and come back so that you can understand and prepare yourself for the test question. So coming to the test question, look at this guys. These are different pairs of molecules which are given on the screen. They're asking you to identify which pair is an enantiomer. So for that you have to recollect what are the different properties of enantiomers. Enantiomers have asymmetric carbon and hence they must be mirror images and they cannot be superimposed. Superimposed you understood from the video. So if you straight away try and look for the mirror images, then many of them will satisfy that property. So please don't go by that. You have to have all 3 properties in an enantiomer pair. First of all, first pair A, do they have asymmetric carbon? No, because they have 2 hydrogens, so no. Coming to the second pair, which is B, it has NH2, CO2H, CH3H. So there are 4. And same thing here, CH3, NH2, H, CO2H. Hey guys, remember we talked about ball and stick model? In ball and stick model, we are trying to show the molecules and their orientation. So do not read this molecule here, this little group here. as H3C. This is not H3C, this is CH3. Why am I reading backwards? It's because that is where it is connected to asymmetric carbon. You have to read the molecule from the point where the bond originates, not where the atom ends. So yes, the pair B, they have asymmetric carbon. Let's see, C, no, they have two hydrogens, so then they do not satisfy the rule for becoming enantiomer pairs. D also does not, because this is CH3 and this is also CH3, guys. So this is not even asymmetric carbon. So asymmetric carbon not there, asymmetric carbon not there, no asymmetric carbon in this pair, no asymmetric carbon in this pair. H and H, it might be easy for you to identify and say that they are not asymmetric carbon, but here guys, the reason why I considered this as not an enantiomer pair is because they do not have asymmetric carbon. Why don't they have asymmetric carbon? It's because they do not have this, 4 different groups attached to them, because this is CH3, this is also CH3. You have to read this one backwards from the point where it is attached. So CH3, this is CH3, this is CH3, CH3. Okay. Enantiomers have application in drugs. When pharmaceutical industries are making drugs, they have to make sure that they're making the right isomer, and the reason why they have to pay attention to this is because one isomer could be crank, the drug which is sold on the roadside, and it makes us a little lethargic and something else, and the other isomer or enantiomer is actually used as medicine for asthma or relief to have proper breathing. So watch out for those guys. Those are the good examples. So yes, pharmaceutical industries are dependent very much on understanding this concept. Next section is on functional groups. What are functional groups? Functional groups are those molecules which are attached to the carbon skeleton, and they give them the properties of being a sugar or protein or DNA or lipids or vitamins, things like that. And functional groups, they are very important for a chemical in its function and how they react, and also give shape and structure. Remember, in molecules, shape is everything. So look at this, guys. This is estradiol and this is testosterone. Look at their molecular formula. This is molecular formula of estradiol. This is molecular formula of testosterone. Do you think they are isomers? I'm just trying to club the two concepts that we learned earlier. That is isomers, molecular formula, with what we are learning now, functional groups. Remember, the rings are the type of carbon skeleton. As you can see, both estradiol and testosterone molecules, one is female hormone and the other one is male hormone. They both have same carbon skeleton. They have 4 fused rings, 1, a 2, a 3, a 4. They both have the same kind of, almost similar kind of carbon skeleton. But look at the molecules which are attached to this carbon skeleton. They don't exactly match, right? So the slight difference of this having this little a group of 4 atoms and this group of 1 atom versus 2 atoms here, that makes one molecule a female hormone, the other one molecule a male hormone, right? So these functional properties, which are the tail molecules which are attached to the carbon skeleton, they play a huge, they make a huge difference. They play a major role in giving the specific properties and in their functions to the molecules. So question, testosterone and estradiol, are they structural isomers? First of all, they don't have same molecular formula, so no. Are they soluble in water? Guys, the rings have nonpolar covalent bonds in them, so they are not quite soluble in water. And are they enantiomers? No, they're not even isomers, so they cannot be structural and enantiomers. None of them, no. Are they proteins? And they do not come under the category of proteins. We will know more about this when we go to the next chapter. So yes, answer is D as in David. Yes, they are both lipids. That's the only possibility. What are the different functional groups? There are 7 of them total, and these are the names: hydroxyl, carbonyl, carboxyl, amino, sulfhydryl, phosphate, methyl. The questions, the kind of questions that you would see in the MCAT test and in competitive exams and in unit tests coming up, and AP classes, biology classes, especially in this class, the coming unit test, the kind of question it might ask. Identify the functional groups. They give you a structure and they give you a bunch of names. Is this methyl? Is this carboxyl? Is this carbonyl? Or is it something else, because there are 7 of them? So just giving you a sample question so that you have an idea when I'm going over the next slides, what you need to look for, what you need to learn from them. Okay, guys, let's move on. So this is the picture from your textbook. As you can see, they gave you the name of the functional group at the very top and the structure. What you need to learn is this purple highlighted part. Yes, you need to know which functional group has what kind of structure. You have to know this very well. And you know its name. You don't have to memorize the structure of the example molecule and its name. Yes, you have to know the properties as well, guys. So that is what you need to look for. Okay, let's go about each functional group. Hydroxyl functional group. What is hydroxyl functional group? Hydroxyl functional group is made of 1 oxygen attached to hydrogen, and oxygen can form 2 bonds. So this oxygen is attached to carbon skeleton. The carbon skeleton can be as simple as 1 carbon or 2 carbon or more than that. So this is hydroxyl functional group. Dash, which indicates a covalent bond, single covalent bond only, and then OH, oxygen attached to hydrogen. Where is it found? It is found in alcohols, and all their names, their chemical names, end in OL. Names end in OL, like ethanol, methanol, propanol. And these molecules are very much soluble in water because of this electronegativity of oxygen, and H can be lost, and then it attracts water or water-like substances, polar substances. So pause the slide and answer this question. Yes, alcohols dissolve in water. Coming to the next functional group, carbonyl. Carbonyl is made of carbon attached to oxygen by double bonds, so it is left with 2 more bonds, and these 2 bonds have to be single bonds. And when this carbonyl is attached to carbons on both ends, on both sides, then it is called a ketone. Whereas this carbonyl is attached to hydrogen on one end and carbon or chain of carbon atoms on the other end, they are called aldehydes. So carbonyl functional group is found in both ketones and aldehydes, both ketones and aldehydes. Guys, remember that ketones and aldehydes are found in sugars. Okay, and when they're found in sugars, they are called ketoses and altoses. Acetone, you might have heard this name. Acetone is a chemical which is found in nail polish remover. The reason why acetone can erase nail polish from your nails is because of this functional group. Remember that. Okay, coming to